Hi, everybody. Welcome to my talk here. And this is Pacific Northwest National Laboratory in Richland, Washington, and Eastern Washington, where it's in Richland in the Tri-Cities, where the Snake River and the Columbia River and the Yakima River come together. And there's 4,500 employees, and it's a Department of Energy research facility that is run on behalf of Battelle for the Department of Energy. All the national labs are run by contractors. I actually work for Battelle, a very large research organization you might have heard of. So today I'm going to talk to you about how we've been using LabKey in the area of developing biofuels and bioproducts. And there are some certain parallels to clinical work, and time series data are especially important to us in these processes. So some of those research that the Department of Energy supports through their Bioenergy Technologies Office. Here's some examples. In Iowa, there's Project Liberty, which takes corn stover here, which is what's left over from the plant when you don't have the corn cob, and it's actually composed of 60% structural carbohydrates. We can take those sugars and feed them to organisms and make fuels. In this case, they're making ethanol, cellulosic ethanol, which has a much lower carbon footprint than corn ethanol. And so ethanol has some properties. It's important. It replaces MBTA, which was a additive to gasoline that caused a lot of groundwater contamination. So ethanol is actually very good for the environment in that way. Another great example here is Lanzatech. They, with the support of the Department of Energy, developed a process for taking steel mill flue gas, which has a lot of carbon monoxide, and bubbling that flue gas through a big column of a culture of bacteria that make ethanol also. And then PNNL has a catalytic process that upgrades that ethanol to jet fuel. And something I've been working on with a team called hydrothermal liquefaction, that's where you can take a variety of wet biomass substrates, sewage sludge, manure, algae, and run it through a hot tube reactor at like 3,000 psi and 350 C, and in the space of about 10 minutes you get out crude oil, basically. So we just sort of take what happens underground over millennia, epochs, and speed it up and make crude oil. And then we can upgrade that via hydrotreating to diesel and jet fuel. So we got interested in LabKey. I got interested in LabKey mainly through my own project work, but I'm also part of a consortium called the Feedstock-Conversion Interface Consortium. So these big plants, especially the biomass plants, like the cellulosic ethanol plant that I showed you, have a great challenge in handling solid materials. Solid feedstock handling is a very challenging approach if you try to get something like salt out of your shaker or something, or ketchup, not ketchup, but things that are solid. And so what we're looking at here is a schematic of a feedstock processing, two different process development units, one at Idaho National Lab and one at National Renewable Energy Lab, for taking bales of corn stover here, grinding it up, and then making a flour that goes into super sacks. It goes off to the National Renewable Energy Lab, where it's further chemically and thermally pre-treated. In some cases, they have a new process that's just all mechanical treatment that's very exciting, and then goes into bioreactors for making fuels. But you can have these problems where the biomass gets stuck and plugs up, and so it's a very challenging problem. So these 8 national labs here, Argonne, Idaho National Lab, Berkeley, Los Alamos, National Renewable Energy Lab, or NREL, Oak Ridge, and us at PNNL, we're all part of this consortium working together to solve these problems. And so what we've done here is that we have LabKey Professional Plus running on the Amazon cloud on a Linux platform. And then at some of the national labs, we have Community Edition running as well. And that was really a great way for the labs to get involved with LabKey. Each national lab is a separate legal entity run by a separate contractor. So we have to do a lot of advocating and getting people on board in this consortium. So the people aspect is extremely important. So allowing people at their own national labs to have their own version of LabKey within the firewall, within which they can keep their own raw data before uploading to the data hub, that was a big selling point. So here our consortium is made up of eight tasks, of which each has several subtasks. And I'm the lead of the data integration and quality by design. Some of you in drug development may have worked with the quality design paradigm within the FDA's guidelines. So we've been trying to adopt that and use that for biomanufacturing. So this is the first task, materials of construction, which looks at the equipment that process the biomass and how the biomass may wear and tear and so forth on that and what they're made of. And you know, the mechanics of wear, things that cause plugging. So over here's a LabKey assay where they correlated. This was from Oak Ridge, and they have what is called energy dispersive spectroscopy, I think it is, or emission dispersive X-ray spectroscopy, and coupled with scanning electron microscopy, where they can get the chemical composition, the elemental composition as they go along, scanning these. So this is in LabKey. There's the nice thumbnail capability that you're all aware of. And you can associate the images with their chemical composition and see that these are mostly silicon, they're little pebbles, but over here we've got some biomass, which is mostly carbon and so forth. Another task is the feedstock variability task. This is mostly led out of Idaho National Lab. And they have a method here where they're looking at a visual image of the biomass and looking at just looking at the RGB channels of biomass. And as it's going through the system, if they see a change in color, you know that maybe something's coming that's going to give the system a bit of indigestion. And so they can have a machine learning algorithm with a video camera and so forth. So that's helping drive this research. And here that's been taken into a study where they've integrated assays from other offline measures to correlate with the visual data, with the RGB data. So there's a whole lot of analyses at multiple scales. And also here, we're trying to identify critical material attributes, and that's a word within the quality by design paradigm, where you understand what's critical about a feedstock going into a process, and then what are the critical process parameters of that process, and then what are the critical quality attributes of your product. So if you think back to this big schematic that I showed you of these big large biorefineries, we have to work backwards from the product and figure out, okay, what's the critical path here that's going to help us keep the plant up 24/7 all year round. And INL also has the Bioenergy Feedstock Library, which is a specimen archive. And what we chose to do is use LabKey in a way where each of the distributed groups across these 8 national labs can generate GWIDs, and then with a report generated within LabKey, update this centralized archive that's already existed. It's built out a custom web application in SQL Server. And it has extensive lineage capabilities and so forth. So taking these legacy platforms and integrating them, I'm sure for all of you, we were just in the talk about organizational adoption of LabKey, how important it is not to try to go in and displace things that are working well. And this is a case where we're moving along that path. Materials handling, they're also doing some things, discrete element modeling of particle flow, and a number of studies for that. And again, another assay. This shows the surface composition measured by nitrogen adsorption onto a tiny particle. You can measure the surface area quite accurately. And again, joined with other data that was collected on the oxide composition. Preprocessing, that's where things are being ground up and put into the system. Then one here is pyrolysis. Pyrolysis is another conversion technology where you take biomass, mainly dry biomass, and you heat it up in the absence of oxygen, and then what comes out is an oil phase, and then there may be a char phase. Maybe you've heard of biochar if you're interested in climate change and carbon management. People talk about biochar. It's made through this process of pyrolysis. But depending on how you tune the process, you can make oil or more char or so forth. So NREL does a lot of this work, and this is actually work that's shown here that was generated at NREL, but then the oil was sent to our team at PNNL where we do hydrotreating, and that's where you replace nitrogen and oxygen with hydrogen to increase the energy content of the oil so that it can be used in diesel and jet fuel and things like that. And then here's the low temperature conversion that relates to what I showed to you with the ethanol plant, but also it has to do with using microorganisms in bioreactors or fermenters to make the fuels from sugar feedstocks typically, but also there's an effort to also try to use the lignin in the plant. The plants may be anywhere from 15 to 20, 30% lignin, and lignin's a very challenging substrate, so there's work going on that on microbial production of fuels from lignin. But it's mostly the sugars. And so there's probably a lot of folks here, maybe some folks from Alder, who are making drugs in bioreactors. If you're making proteins, drugs, therapeutics, monoclonals, you have people with bioreactors. I work personally with bioreactors in a lab. I do lab work as well. And I'll talk a little bit more about this. So we're really interested in seeing the overlap between the biopharmaceutical sector and industrial biotechnology, and seeing how together we can work together to bring in data into LabKey. When you get to the industrial level, you have systems for process control. They may have heard of SCADA systems, supervisory control and data acquisition. That's where in your big plant you bring in all the data and you control pumps and things from a central control room. Those platforms are set up typically for continuous operations and not necessarily batch. And also they may have a LIMS off to the side. But what's different about our endeavor is that we have this whole research component where we're making up experiments to study the system, and we need to integrate all that. And that's where LabKey really is so well placed. And also the fact that LabKey is based on a broader scientific method instead of just hard-coded for a specific use case has really allowed us to take it and apply it in this very different domain. And there's going to be multiple examples that I think we'll see over the years. One of the folks in the consortium from Oak Ridge National Lab, Stan Martin, he's leading a separate consortium, the data part of a separate consortium. And they're looking at plants as feedstocks for biofuels, and he actually has trees and plants as his patients. And on his LabKey platform, you can go to his website and bring up a tree garden in Oregon and identify his tree patients. And so they're studying the genomics of these trees and so forth, and how we can engineer highly efficient plants to produce carbon neutral fuels. So again, some of the considerations here we'll talk about, on porting LabKey over to this domain. But one of the things, again, biomass deconstruction is kind of like mammalian digestion. We don't want our plants to get an upset tummy and just sit down and stop working. So we try to keep them happy by understanding what they're feeding, how much we can feed them, at what rate, and so forth, so they don't go belly up and cause us to have a bad day. So here, some of the considerations that we have to think about with these big operations, in great big biochemical plants, biorefineries we call them, is who's the subject? It's easy to think about a subject as patients, but is it the subject our feedstock all the way through as we're doing studies? Or is it each of the individual unit operations? Should we call those things subjects? So we've only been going with LabKey really full on for about 5 months. So we're having to talk through a lot of these issues on a broader scale, and so that we get it right from the outset with our paradigm of how we're going to handle the data. So also aligning the timestamps of different unit operations, that's important. So coming up, and I met Liz there, she's got at the Hutch, they're doing a cool thing where they're managing their equipment laboratory through LabKey. So if you have all these processes, you have to have a unified start time where you can relate the sample data and the process back to some overall, let's say digestion, okay, at 10 o'clock I ate a bagel, and then, by 10:02 it was in the stomach, and you know what I mean? So if you want to align these data, you have to have a uniform time base. Windowing functions are something that are very important that we'd like to see in LabKey. I'll talk a little bit about some data where we integrate the data. LabKey SQL doesn't necessarily bring all the functions up from the underlying SQL of SQL Server or Postgres. And so if you want to do window averaging or timestamp matching, very critical, literal timestamp matching, it requires those functions. And so anyway, we're interested, again, looking at the overlaps in the clinical area, especially like ICU work, surgery, and so forth. They all have time series data as well. So coming up with some generalized data structures on the LabKey platform would be an interesting thing for us. So I'm going to talk now a little bit about a experiment that we've been working on in the lab. This is one I'm really interested in, been working closely with these companies. And that's to bring in advanced spectroscopy practices into our bioreactor operations. So our bioreactors range from these smaller bioreactors, maybe half a liter or a few liters up to 30 liters, and we've got a 120-liter one in our lab. And so 3 different modes of spectroscopy that we've applied to infer information about the growing culture in real time so that we can have advanced control systems. One is dielectric spectroscopy, where you have a radio frequency probe that generates an oscillating current in the culture. And because cells are like little batteries or capacitors, you can measure their capacitance of the bulk solution as being proportional to the viable cell mass in the tank. Because a living cell will maintain its cell wall and maintain a dielectric exclusion, but as they die, they get leaky and so forth. So that's something that we worked on early on. We worked on near-infrared spectroscopy real time with Sartorius, and or to be able to develop some process control systems on that. And then over the past couple of years, we've been working with Metrics, which is a spin out of the University of Washington Applied Physics Lab. And they've come out with a Raman analyzer that's a low-cost, high-resolution analyzer, and also a probe with a sapphire spherical lens that has very high optical resolution, but also makes the probe very adaptable. You can stick it in a liquid or a powder or a slurry, and it's instantly in focus. So they're going great and selling these things like hotcakes. So we're looking at applying that to the bioreactor. And so we have started to use LabKey as a chemometric data integration platform. And so here's an experiment we just ran, just finished it a week ago, basically, where we've got this little reactor here, and we have over here a real-time prediction engine that was being used with a chemometric model of the Raman spectra, that we were able to correlate the glucose, xylose, citric acid, and xylitol levels with a specific pattern in the Raman spectra. And so here, what I'm going to show you is how we built this and how LabKey played a role in that. The setup here is that we've got a peristaltic pump that continually draws filtered bioreactor broth from the bioreactor through the Raman flow cell. The Raman flow cell, the analyzer fires every 3 minutes, and the laser generates a Raman spectrum, and that's written to a text file. And there are 1,900 intensities, 1,900 wave numbers in each text file along with their intensities. So those get uploaded to LabKey along with samples that are pulled by a Flonamics auto sampler every 30 minutes to a few hours. And that gives us our reference data in order to train the chemometric model. This is a form of machine learning, of course, where we're using a multiple regression model, a partial least squares model, to predict the concentrations of these components of the bioreactor broth in real time. And so this is what I was just talking about. So we bring the HPLC data. It's a piece of cake to bring in LC data into LabKey and upload it in an assay from a spreadsheet or wherever your person is giving it to you. The other one, we had to write a Python script that would take in these. By the end of the week, we'd have 3,000 individual text files. So I wrote a Python script on the upload side of the assay that would take these individual text files and then put them into a table format. But each time we collect a Raman spectrum, there are about 1,940 individual values. So we found that we could load these into a multi-line text field of an individual record. And it works pretty well. And so those are the individual records within the assay right there. Each one of these is a text file, and not shown here, but shown here are the intensities that go along. So then I had a terrific intern this summer, Isaiah Lemon, who's a double major Chemie and CS at University of Washington, and he coded a great R pipeline for us. And basically what we need to do is we need to join the LC data with the Raman spectrum at the particular time that sample was physically pulled from the bioreactor and then measured for its chemical components. So given that the Raman system fires every 3 minutes, takes a reading every 3 minutes, but we only were pulling LC data every 45 minutes, we had to go in and time stamp match. And that's what his script does. It loads those into data frames in R's and then uses a windowing function within R's. And it generates a text file here that you can download. And this is just one of the Raman reads right here. File goes on for a long way. And then we took it into infometrics Pirouette. Pirouette is a chemometric modeling platform, just like Umetrics Simca or Unscrambler or the PLS2 locks in MATLAB for doing chemometrics, for doing multiple regression modeling and for classification. Infometrics is based out of Bothell. They've been around since the 80s, so they've got a big following in the energy business. And so we work with them on some research as well. So here are spectra, the Raman spectra, as they come off the system. And then here's some smooth and transform data with a baseline correction. And so here, these peaks, like the light blue, that's where the glucose and xylose were high in the culture, and the red, that's where later in the culture when these sugars had been consumed by the yeast in this case. And so if we can zoom in and we can see features like this, we know right away we can build a model. If we can see them, we can certainly build a model. In some cases, even if you can't discern things, you can build a model. So we tried it out the first time just a couple of weeks ago, and it worked great tracking the glucose. Here's the glucose prediction over time, all this sort of point cloud with a moving average. And here's the average. Here's the actual measured LC glucose concentration while we were making citric acid. And this went on, made more up here. But so we were happy with this because for bioreactor work, keeping things within like 0.5 gram to a few grams per liter, that's great. We can go home at night and let it run overnight in the lab. And that's what's really, really important is automation in the lab is so critical now for going forward. Because to get this kind of data, we would have to come in at night and pull samples and things, and it's just not possible. So here's the vision with LabKey, is to not only integrate the Raman data, but also the near-infrared and the dielectric spectra as well, and combine those with the category variables that might come up with other information that you collect about the bioreactor, like what was the dissolved oxygen at that time? What was the pH? What was the CO2 off-gas level? Those kinds of things. And so those category variables allow you to classify your spectra and group the spectra as you are building the multiple regression model. And then the Y block, those are the measured variables that you want to build the model to predict for. And so then once you have the model, Infometrics also sells a prediction engine called InStep that you load onto a computer, and then it controls a PID, proportional integral derivative control loop, to automatically spoon feed the culture the feed stock. In our case, this was corn stover hydrolysate, which is high sugar feed. It sort of looks a little like molasses. A little dark, and it's basically we're just feeding it candy, because if we were to put all those sugars up in front, it would just be an overwhelming osmotic load on the cells. So by spoon feeding them, we get better performance in a fed batch culture. So a goal what we want to build is something we're calling a fitness landscape explorer, because we'd like to be able to use these spectroscopy systems to automate how we control the cultures so that we understand where the optimum performance of the culture is, or we use a chemostat to evolve the culture to a higher performance. One of the challenges when you're doing this kind of direct evolution, you don't know where the peaks of optimality are. It's a huge combinatorial space of the different process parameters, how much oxygen you're giving it, the temperature you're running at the pH and so forth, and the nutritional components. And so there's a great paper that came out a couple of years ago from Stephanopoulos and Lamb, MIT, where they found that ethanol tolerance in yeast is a hard problem when you're trying to make like 200 grams per liter of ethanol, or whatever, or 100 at least, or something like that. That's really hard on the cells because ethanol pokes holes in cells, it solubilizes the membranes. So it turned out that they did some gene expression studies and found that the potassium pumps in these cells were working overtime. And what they were trying to do is balance their potassium levels with the external environment because the ethanol was poking holes in their membranes. And they found if they just adjusted the culture medium to have the same amount of potassium as the cells would like to have inside, the ethanol tolerance went way up. But that had to be found through this whole gene expression studies, So what we'd like to be able to do is robotically explore fitness space so that we can better get to the optimal performance of the microbes and also understand how to engineer them for better performance. So these are some of our issues that we put up with LabKey, and I can talk to you all about this. We're running up against the time, but they've been great. Hannah and our team and John have been great. But one of the things that we're really happy about is now when you copy the study, there's a little button here that says display date time. That now we can see the dates because the continuous studies only had the days, and if we're down to the minute or so, it was upsetting to us that we couldn't see the date. So now it has this function, so you can go there, click on that, bang, it populates with the dates. So anyway, there's our team, and it's been a lot of fun working with the folks at LabKey, and part of this has been getting the other national labs to come on board with LabKey as well. So the community edition again, was a great opening for them to be able to, see that this isn't just a PNNL run thing, that their own national labs can run their own instance of LabKey, keep their raw data together until it's ready for uploading to the data hub. So, thanks.